Okay, thanks for the introduction, Hero. So yeah, it was really tough to arrive here last night, but we made it. And thanks for the opportunity to come here and talk to you about some of the research that I probably should stay here that I have been doing in the past years. So to start off, I'm gonna walk you through this outline that I prepared for this presentation today. I'm going to talk to you about the peripartal period, a brief resume about what has been done in peripartal period, and some of the effects that we have seen with methionine during the transition period of dairy cows. Then I'm going to talk more about liver function and how this relates to inflammation, oxidative stress, and other effects that are really important during the transition period of dairy cows. And finally, I'm going to talk more about this effect that we are currently doing our research on this in South Dakota. Is about histone methylation, but primarily on the effect of methionine at the molecular level. And at the end, I'm going to give you some conclusions and remarks about this, where this is going. So just to an introduction for the peripartal period, we know that there is a time where the cow has a lot of changes physiologically and metabolically. We know that the cow has to adapt to start production, and therefore the cow has to derive or move some nutrients towards this milk production. Therefore the cow has to mobilize tissue, likely fat, but also we know now that it's gonna modify, I don't know if this is correct, mobilize tissue, muscle tissue, in order to supply those amino acids during the transition period. On top of that, we have also the effect of a decrease in intake during those days around calving. So this can really exacerbate the problem of the transition from late pregnancy into lactation. So all this, with all this research, we know that there is a decrease in body condition score in a weight or a weight so the cow can cope with that deficit of energy balance. And because of all these effects during the transition period, we know that cows are likely to develop a disease during this time because of all these adaptations. Some cows might go through this transition period very well, but other ones might not go as smooth. So we have a huge amount of diseases occurring during those days right after calving. So most of you have seen this scheme from the Drakeley's paper. So just to bring back the idea of the transition period or cows that are in negative energy balance, we know that from the fat or adipose tissue, there's going to be a mobilization of fatty acids or NIFA to the liver, primarily because there is a decrease in insulin sensitivity or responsiveness, and also there's an increase in epinephrines, which render those triglycerides, is that state between triglyceride and NIFAs, more towards NIFA, and there's going to be that mobilization of fatty acids to the liver. Once they reach the liver, we know that they have a fate, that they can be utilized for energy by oxidize these fatty acids in the mitochondria, but also there is a partial oxidation that can rise to increase the amount of ketone bodies. Additionally, we know that fatty acids can be packed back into triglycerides and into VLDLs, very low density lipoproteins. There are proteins specialized to move out those fatty acids from the liver back into the bloodstream, and they can be utilized by the mammary gland for milk fat. So all these efforts here, for instance, for VLDL, is to decrease that concentration of fatty acids that can lead to fatty liver. There is another disease during the transition period. But so far I have been talking only about in terms of energy, but also there is another side that Dr. Ferraro was talking about on the protein side during the transition period. There is also a mobilization of protein. And in fact, this is a study where they observe what you're looking at is body fat and protein percentage of the cows between 2 weeks, at 5 weeks, and 12 weeks relative to parturition. And what you're looking at here is a decrease not only in the yellow bars, not only in the fat percentage or amount in the body of the cows, but also in protein, there is also a decrease between 2 weeks and 5 weeks around the parturition, meaning that the cows, again, the same idea, is going to mobilize not only fat, but also muscle tissue to supply those amino acids required. Interesting is that they also put those cows into different state of crude protein, and there still was a significant reduction, same reduction on the body content and muscle. So it's same idea that Dr. Ferraro was talking about in another presentation. So what happened here is that there is a decrease this metabolizable protein, mainly considered by ruminant undegradable protein and microbial protein. And you can see that this is a study from Cornell as well. You can see there is a decrease in the balance of metabolizable protein. So just as negative energy balance, we also have a decrease in metabolizable protein balance during those first few days after calving, around 7 days. And then by 3 to 4 weeks after calving, that balance comes back up to 0. So this is a lot of concern for us, because it's not only about the quantity of metabolizable protein, but also the profile of amino acids that are within this metabolizable protein. So most of this decrease here is because the cow will require amino acids for glucose synthesis, obviously for milk protein, and also for other compounds that I'm going to talk about today. There is SAM, S-adenosyl methionine. There is an important methyl donor for the body, and also glutathione, antioxidants, and taurine is also another important antioxidant. So, as some of the other speakers have said today, the methionine and lysine has been regarded as the most limited amino acids. We know this from the NRC and from other studies, and therefore that was the question that we initially took when we were starting those experiments in transition cows back in Illinois, looking at whether feeding methionine and lysine during the transition period would be beneficial for peripartal or for transition cows in that sense. So what you're looking at here is just another graph, just to explain this liver uptake of methionine and other amino acids during the transition period. You can see that all of these are amino acids, and all of them, they have a significant linear effect that they decrease the uptake of these amino acids by the liver. They decrease during the transition period. However, if you notice methionine and histone, they are the only ones that have a quadratic effect, meaning that between minus 14 days and 4 days, these two amino acids, the uptake actually increase and they decrease again. So there's a quadratic effect going on here, meaning that if you remember the metabolizable protein picture that I show you, is around the same days when the cow requires the most metabolizable protein balance. There is an uptake of these specific amino acids, and perhaps this comes back to the same idea. It's not only about the amount of metabolizable protein, but the profile of those specific amino acids. This is, most of you know about this, the metabolizable protein. Where is it coming from? We know there is 2 portions to the crude protein, undegradable and degradable protein. Most of these degradable proteins will be utilized by the microbes to make up the microbial protein, along with some starch of energy. And also this nitrogen can be recycled back to the rumen. So most of the metabolizable protein that is absorbed in the intestine come up from ruminant degradable protein and microbial protein. And whatever is left off is undigestible protein. Okay, so this metabolizable protein, like I said, is not only about the amount that we are supplying to the cow, but also the profile of amino acids. And as we know, the methionine and lysine was originally tested in milk lactation cows. And that was another reason why we start those experiments back in Illinois by feeding methionine during the transition period before calving. So that's about peripartal period. Now I'm going to talk about those series of studies that have been conducted at Illinois. So here's a table that I put together just to show you, put back to back all those 3 experiments. You can see they have fairly similar crude protein percentage. They also, the supply of metabolizable protein is slightly different for one of the experiments, but at the end, the bottom line here is that most of the lysine to methionine ratio is around 2.8, 2.7. And you can see the contrasting difference within the control. So these are control for within each of these experiments. Okay. So from this, we observe a pretty consistent effect by feeding this rumen-protected methionine, a pretty consistent effect by increasing meal yield. We observe around 3.4 kilograms in the study that I run, and then subsequently we have observed an increase in meal yield 3.8 and 4.2 kilograms per day. Even though the last one experiment was they go up to 60 days, days in milk, and you still see an effect, by feeding rumen-protected methionine. Also, we observe in the case of intake, we also observe a consistent effect by increasing intake in those cows starting the lactation, around between 1.6 kilograms of dry matter intake to 2 kilograms of dry matter intake. Same idea is pretty consistent across all these experiments. In case of milk protein, we also observe an increase in the total milk protein for all the 3 experiments. And we also observe, in the case of one of the experiments, we observe effects on milk fat as well. So what I'm trying to say here with this sequence of data that I'm showing you is that we have seen this quite a lot, this analogy. There is the iceberg. The tip of the iceberg is what we have been seeing at the level of milk yield and performance, intakes, components in the milk. However, through this research that we have done, we have been able to observe what is going on in the background, down below at the level of the metabolites or metabolism of the cow, and also at the molecular level, how these effects can take place and adapt the cow, or start a cow with a better status to start the lactation. So now I'm going to talk about the liver function. We have seen time after time this is a very important effect to keep a good start for to put the cow in a good start for the lactation, to start the lactation. And methionine we have seen a lot of these effects with this research. They can interact with the 1-carbon metabolism and the biosynthesis of DNA, RNA, and also for the synthesis of VLDL, that molecule that I showed you before, to increase the apolipoproteins, and also for phosphatidylcholine. We also have seen at the molecular level some effects with DNA methylation, and now what we are researching in South Dakota is histone methylation. Obviously, there is a methionine as a precursor of cysteine. It also has an effect on this. By increasing cysteine, you can also have down the stream, an increase in those antioxidants, taurine and glutathione. With this, increasing the antioxidant capacity for the animal, especially during this transition period, it's quite important to have this. Not only to ameliorate the increase in milk production, you're also going to increase oxygen radicals just by having a higher metabolism, but also to help those immune cells that are trying to combat any potential pathogen due to metritis or mastitis, and consequently reduce the immunosuppression that occurs during the transition period as well. So for us, we look at liver function in a way there is a balance between inflammation and oxidative stress. We know that oxidation is not something bad, but it's a problem that is something that continues, for inflammation there has to be an initiation and then a resolution. If that doesn't happen, then we have problems because we are utilizing enough additional nutrients to supply this inflammation. And also oxidative stress is another source for utilizing nutrients when we don't really need. So the higher liver inflammation, then you're going to reduce, the higher the liver function, you're going to reduce your inflammation and oxidative stress. That's the whole idea. And this can happen, by genetics. Some cows will have a high liver function and do not require much, they're not going to have inflammation or oxidative stress, and they're going to go through the whole transition period pretty well with no problems. Management as well, and what we're trying to affect here is the nutrition through the balancing of the amino acids. So this is another graph just to show you where the methionine have an effect within the liver. We have observed that methionine, as I mentioned before, it can increase that concentration of glutathione and taurine in the liver. It can also serve as a source for methyl, for choline, phosphatidylcholine, that is an important component of the VLDL formation, and to export those fatty acids that normally arrive during the transition period because of the low energy balance. Another point here is for methionine is for the synthesis of apolipoproteins, ApoB. This can substantially affect that other component that is required for VLDL, primarily because they need this protein, so once it's exported outside of the liver, other tissues can recognize, okay, so this is a VLDL, we're going to use this molecule. Then the other effect is with albumin. We have seen very consistent effect of methionine on albumin during the transition period. Albumin is a major secretory protein from the liver, but also is a major carrier in the blood. So all those NIFAs, likely they are being carried, to some extent through with the use of albumin in the body. So now I'm just going to show you some another. Not going to go into much detail on this, but it's an effect that we have observed by feeding methionine to transition dairy cows in terms of carnitine metabolism. So if you remember, there are a lot of NIFAs coming, because of the fatty mobilization to the liver. So the liver has to utilize those fatty acids in the mitochondria. So this carnitine protein, what it does is allows these fatty acids to go across these two layers between the cytosol in the cell and the mitochondria inside. So if the fatty acids remain here in the cytosol, they're going to accumulate, and we don't want that because that leads to fatty liver and other is a negative effect on the liver function at the end. Okay, so by having this protein here, we're going to move all those fatty acids inside of the mitochondria, and they can be utilized for energy. So what happened is that we have seen that lysine can be trimethylated into 3-methyl-lysine, and this component is one of the rate limiting points in the de novo formation or synthesis of carnitine. Okay, so by having lysine and methionine in the diet, you can also affect that concentration of carnitine in the liver. So consequently, you're going to have more chances that those fatty acids can go across between the cytosol and the mitochondria and be utilized for energy. So these are the results on that. You can see carnitine, there is an increase, especially at 7 days, when you feed Metasmart and Smartamine in comparison to the control. Not so much on day 21, but I think that this is really crucial, because if we go back, those days, day 7 is when the metabolizable protein is much lower, nadir levels, and you're also going to require those days to produce more of this carnitine so it can be utilized for energy fuel. This is another effect from ApoB. This is the apolipoprotein that I mentioned before. So this is a picture of the VLDL. So this is the ApoB protein that is in the surface of the VLDL. And also you're going to have all the triglycerides. They are the fatty acids that are packed into the VLDL. So what we observed here was a trend in our study for an increase in the concentration of ApoB100 during the transition period of these cows. Then Sun recently also published this paper where he observed an actual increase in the concentration of ApoB during the transition period. And he also observed an increase in the concentration of VLDL in the blood. So it's pretty consistent with our theory that methionine can increase the ApoB100 concentration or the secretion of VLDL from the liver and consequently alleviate the fatty liver conditions. This is another map. Is a little busy, but the main idea here is just to show you what is the inflammatory response and how we have come about to understand what those biomarkers of inflammatory response means. So whenever you have an effect or a potential inflammatory response at the local level for mastitis or metritis, you're going to have several signals sent from those local sites in the form of cytokines, and they're going to go into the liver or the brain. They're going to produce a secondary effect of increasing the levels of adrenocorticotropic hormone, that this is going to go to the kidneys and produce cortisol. So it's the stress hormone. So we have stress going into the liver, that signal, but also they can go straight into the liver and produce that acute phase response. Okay? So once the signal reaches the liver, the acute phase response, what it means is that the liver normally produces a constant amount of proteins or a profile of proteins, but when this happens, you're going to have an alteration in the normal synthesis of those proteins that the liver is producing. So that's why the proteins that are produced or synthesized in a greater amount during an inflammatory response, they are called positive acute phase proteins. And the ones that are produced in a lesser amount, those are negative acute phase proteins. So obviously, you can see that apolipoproteins are negative. So if there is inflammation, the liver is going to produce less of this. And also the albumin, there is another important protein that is secreted by the liver. These are other serum amyloid A, haptoglobin, ceruloplasmin. Those have a specific effects within the body, but we know that these are positive acute phase proteins as well. So in other terms, when you have a metritis or mastitis, that's a signal going from those sites to the liver and tell the liver, hey, there is something going on. We need you to adapt what you're doing. Whatever you're doing, you have to change because we have some problem here. So then the liver get that information and then start to producing this change in the synthesis of proteins. So if we put together this, you can see how the liver is really overwhelmed during this time. He has to deal with these inflammatory response signals. He also has to deal with the amount of fatty acid that occurred during this time. He has to do gluconeogenesis. So it's really busy, it's really packed. So those are the problems that when improving liver function is essential. It's a key role for a good transition during the transition period of dairy cows. So just keep that in mind, what is going on there. So right now I'm going to show you some data on albumin so you have a better idea about this biomarker. These are concentration of albumin during the transition period. This bottom here is the control, and these are the methionine, Metasmart and Smartamine cows. And you can see that there is a drop in albumin during those days. Same days, 7-14, they decrease in the concentration in the control. So just this line here is just a reference to this other paper from Bertoni. They have worked extensively in this area of inflammatory response and biomarkers. And you can see this at same 35 grams per liter. This is data from a retrospective analysis where they separate. They have a bunch of cows during the transition period, and they separate in 4 groups. Groups that have a high liver function, medium high, or intermediate high, medium low, and low liver function. So you can see how the concentration of albumin just drops during those days as well when the cow has a low liver function. And just to give you a reference, how important is this? between the high group and the low group, there was a difference of about 6.5 kilograms in production. So you can tell how important it is to maintain this liver function, and this biomarker can tell you a lot about this. And we also, similar effects have been observed for the other two experiments run at Illinois. Now let's look at the positive acute phase proteins. This is a little less intuitive because they increase, but you want them low because the more they produce, the more effect the liver is going to be. So in this case, we have ceruloplasmin and serum amyloid A. This is the control, and Metasmart and smartamine. You can see a decrease in the concentration of those cows with both of these biomarkers for inflammation. So without a doubt, we know that there is an effect of methionine in the inflammatory response. We know that the other experiments also, they have similar effect. So liver function is really important. You have seen how it can damp the whole milk production of the cows. Now I'm going to talk about these other effects at the level of antioxidants. So what you're looking at here is, again, control Metasmart in both. This is ORAC, or oxygen radicals absorbent capacity. It's a long name to say the amount or the capacity of antioxidant, or the antioxidant capacity of those cows in the blood, this here. And this here is the amount of glutathione. In that case, you can see there is an increase in the antioxidant capacity of those cows, primarily in Metasmart, and also we observe an increase in the concentration of glutathione and an antioxidant in the liver of those cows during the transition period. Now, an effect that we observed here was this increase in glutathione in the days before calving, around 10 days. To us, more and more, as we're looking at this data, we know that this is an important effect that can affect not only oxidative stress, but also liver function. Because we believe that there is a priming effect of glutathione by feeding methionine prior to calving, and this goes related to the data that Dr. Ferraro was talking about, the feeding cows before calving. So we have seen in the other experiments as well an increase in the concentration of glutathione in the liver, pretty consistent. And the main point here is that glutathione is not only for a good antioxidant, but also from rodent data or human data is really considered as an amino acid storage in the liver. So what you're looking at here maybe is a reservoir of amino acids prior to calving that the cow can just draw from there and utilize that rapidly between those days, between the change and start lactation. The cow can cash out on that concentration of glutathione. And also glutathione is one of the main sources for cysteine to the mammary gland, so it comes back around to the utilization of this, potential utilization of this for milk proteins during the first few days after calving. Now let's talk about histone methylation. So for us, this data help us, the ones that we work with nutrionomics, it help us to understand methionine as an example of nutrionomic effects, okay? So we're a lot to get this question when we start talking about nutrionomics, because nobody knows nutri-what. So I'll try to explain in a few slides before I move on with whatever the histone methylation I want to talk about. So most of you might be related to genome testing, in cows. From this company, they run genome testing, so it's more known in their industry to know about this. So what you're doing, and it's a good tool to know the genetic traits in your herd. And what you're looking at here when you do this genomic testing is you're looking at DNA, the information in the DNA for those cows. So that's an important part of how the metabolism will work at the cow in a molecular level, but it's not all the information because that information has to be utilized a certain way, and it can be altered by the different type of diets they're feeding. So another way to explain this, because I don't want to go in pathways and kind of things. So how many of you like tiramisu? Okay, it's better last time that I asked this. So, so how many of you can make tiramisu? Very few. Okay, that's better. So those of you that raised their hand, pretty sure if I give you this recipe, you're gonna be able to make a tiramisu. Not like this, but maybe something closer, okay? But what happens if I give you this recipe in Spanish? That's gonna be tricky, right? So it's exactly the same information, the same recipe, but you're gonna have problems figuring out what is a bizcocho, or yema, So what is going on, if we put aside, scientific correctness and whatever, this information is the same information, but in a different language, okay? The DNA is in Spanish, the RNA is in English, and these guys here, if you look at this, these are proteins that make other proteins, but these proteins can only speak in English. So that's the main point here. So if you give them something in Spanish, they're not going to understand this. So, and at the end, they're not going to be able to produce that protein or that final product. So if you're seeing this, these are flux of information, just in a different presentation, but at the end you have to have the same end product, okay? So what we're working in nutrionomics is to understand this flux of information and how nutrients can affect that, because there are a number of regulatory mechanisms for that, and that's what we're studying. So I hope that come across. And for instance, here I have a nutrient that can interact with the DNA, but it doesn't interact directly. It needs a middleman, a mediator. So those, normally we call them transcription factors, is a fancy word for that. And these transcription factors are going to actually go into the DNA and change the amount of, if you remember the recipe in English, the RNA, that's going to affect that. And at the end, all that can roll, all the way to change the performance and also the health and also the performance of their cows. So at the end, we're looking at a nutrient-gene interaction. With this, what we're trying to make an effort, make conscious, among their industry, is that the TMR, what you're feeding the cows, is not only the ingredients or these nutrients in the diet, they're only the building blocks for the cell, tissue, and organs, or they can be utilized as a fuel for the cells, but also they are bioactive molecules. So the cells can actually sense the availability of specific nutrients and responds to that. So going back to that figure, we have the meal yield performance. We also have the effects on liver function, antioxidant stress. This primarily the metabolism of the cow. And at the very bottom, we have an effect on gene expression of RNA, or the recipe in English, the middle one, okay? That's what we're after, and that's what have been affected by methionine. So for instance, to give you an example, these are microarray data from the study that we run at Illinois. What you're seeing is each of these lines is a recipe. And we observe around more than 2,000 genes affected by the supplementation of methionine in the liver. So you can see how contrasting this profile of genes are between the control and methionine. And we already have the ability to understand what major pathways they are affected. And we know that this, what we don't know is how this occurred. If you go into the nutrient-gene interaction, we still don't know to what extent that's happening. But we know for certain there is an effect. And like this, there is many other nutrients that have been observed to cause a change in the gene expression. So because of that, I have developed this model. It's very rustic for now, but this based on some of the review papers that I have read and other data that I have collected. So at the end, I just want to understand how methionine can cause that effect or that alteration in the gene expression. So we know that, for instance, an intermediate metabolite is cysteine. Cysteine can interact with some middleman transcription factors and this transcription factor in a way can go into the DNA and change that gene expression. There is some data showing that some amino acids can have an interaction with a transcription factor. And also there is all this array of information on cell membrane transporter that we just barely know how they work, but not in the sense that they can transport amino acids, but they also can sense the amount or the availability of specific amino acids. So that can send a whole new signal downstream of the cell to cause that gene expression to say, oh, there is a lot of lysine, so we need to reduce or make this adjustment down below at the gene expression level. Now, the other effect that is very particular with methionine is because it produces or is the primary source for SAM, S-adenosylmethionine. There is a methyl donor in the body. So this can be a source for DNA methylation and histomethylation. So I already did a research on this DNA methylation. We know that there is an effect on global DNA methylation and also can affect very specific genes and affect the transcription of one specific transcription factor. So we know that there is an effect here. The other thing is that we know that a lot of times people says, no, methylation is bad and we shouldn't have, but in reality we don't know that much because there are proteins that demethylate the DNA. There is a lot of interactions that happen here. And in fact, this is a very non-intuitive what we observe because we have an increase in global DNA methylation. So overall the DNA was methylated, but then we observed a decrease in the specific regions of the DNA with methionine. So it's like an alter effect. While the whole DNA was methylated, very specific regions for specific and important genes were demethylated with the supplementation of methionine. And now what we are looking at in South Dakota is looking at histomethylation. So just to give you an idea, histones are proteins in the cell where the DNA spools or winds around and create these pockets or pack. Is a very packed around, the DNA packs around these proteins. And with that, it doesn't allow, if it's so packed, it doesn't allow for that transcription or translation of that information. Remember, the recipe in Spanish, it cannot be translated into English when it's like this. But then when these histones unpack the DNA, it allows that information to be available for translation and transcription. Okay? So there are many effects. Acetylation, methylation is just one of the histone modifications that can occur in the DNA. And if you go back to the tiramisu analogy, if the tiramisu is an item in the menu or restaurant, then the whole menu you can consider as the whole genome. Okay? Then what histomethylation is doing is blocking some of those items in the menu. So you cannot see the whole menu, you know. And that's what it's doing. And it's not stable, it's dynamic, you know, based. We know for some research before that the whole status of histones might change between late lactation and will change throughout the whole subsequent lactation, primarily to allow a stable, let's say, a stable menu throughout the whole lactation. So you produce the same amount of caseins, kappa caseins, and all that information is available. So you don't want that to be changing around during the whole lactation, but it is start changing as the cow moves into late lactation. So what we have been doing here is we're utilizing modified histone protein that was developed in MIT. And what we're doing is literally shooting this information inside of bovine cells. So we're putting that recipe inside of the bovine cell, and the bovine cell will not distinguish if it's in English or it's gonna make the protein. So these are bovine cells that we can keep in this microscope, and we can follow them as they are producing these proteins. And if you can see, this is the histone peptide, and there's a place there where we can, it serves like a bait for those methyl enzymes. So the methyl enzyme will recognize this as a histone, so it's going to methylate that, and it's going to increase the specific intensity of those cells. So what we're doing with that is not only to go there and look at how cool they look, those cells, but what we're trying to do is to move that qualitative data into quantitative data. And what we're doing is to utilizing this cell profiler, is a free software online. What it does is a pattern recognition software. So it recognizes patterns with a background, and it will tell you how many cells there are, what are the shape, what is the intensity, that's what we are after, and a lot more information that we don't really need, but we need that intensity. So the more intense the intensity of those cells, the more methylation you're having, basically. So so these are an actual treatment that we run by increasing the concentration of methionine in those cells. And you can see that the intensity is quite different between the treatments. However, there is not a dose-dependent manner. We run this several times, and time after time, we saw an increase with the 125 micrograms. So if it's not a dose-dependent matter, it means that there is something else, within this biological process that we're not accounting for. However, we proved that, in fact, we can increase methylation when methionine is present in the media of those cells. Okay, so there is, in fact, an increase. As we speak right now, we're running other data on this, global DNA methylation, genes, and see how these pan out to have a whole story of what this means in terms of the physiology of the cow. And this is what we're going after. This is proposed hybrid experiments in which what we're going to be doing is to provide TMR with different concentration or different ratios of lysine methionine to those cows, then get the serum from the cows at several time points if we want, and then incubate those cells with the serum from those cows. So and these cells have been preloaded with a histone, modified histone that we use. So what we're trying to do here is combine the on-farm variation, the environment, animal variation that occurs here with the highly controlled environment of in-lab assays. And in that case, we can actually account for or get a more tangible effects, not only at very controlled environment, we put methionine and okay, this happens, but also we can have the ability to account for animal variation and the other things that I mentioned. So at the end, you can have a histomethylation effect based on this, but what we really are after is that this technique is cheaper, than running a PCR, and also can give you a lot of data. In one single run, we can have a lot more data, and we're going to be able, at the end, that's the goal, is to make prediction models. Predict, with this ratio lysine to methionine, we're going to have this amount of histomethylation. And furthermore, we're just beginning with this because then you can see or trying to understand which regions of the DNA are actually opening up or closed based on different ratios. And also, I want you to understand that this is just one example. So this can be utilized with other different techniques, and especially if you are using fluorescent proteins as well. So just to close this out, these are the final remarks. So what we talk about first is that without a doubt, we observe an effect of methionine at the level of performance, then metabolism, and then gene expression of the cow. And these allow those cows to have a better liver function and have a smooth transition into lactation by controlling, or having a better control of the inflammation that is going on and the oxidative stress, and with that improving the performance. Then the extent of this histone methylation that we're running right now, we don't know. At this point, we will have more data pretty soon to know what exactly does this means. But certainly this can add up to the methionine model that we're trying to develop and see how this is an example for a nutrionomic effect for other type of nutrients in the future. And with that, I'll answer any questions.
I think we have time for one question. Oh.
It seems that the goal of feeding methionine is to increase methylation, histone methylate, right?
No,
Is that necessarily a good thing? That's kind of my question.
Yeah.
So the dogma is changing a little bit, but aren't you risking maybe silencing some genes that are in fact important for patient performance?
Yeah, so the goal is not to increase the methylation, but rather understand what sections of the DNA, because if the DNA is packed in one side, it's very likely that it's going to be unpacked or available in some other regions. And the fact that it's not a dose-dependent effect means that there are other things going on that we don't know the whole picture of how this biological process works. But definitely, that's something that we want to understand better. What does it mean, at the end? Can we connect this cellular effect with the overall performance? Can we, at will, at some point, unlock sections of the DNA that we want? Can we unlock sections for proteins that are good for human health? Those effects, we don't know at this point, but we are starting with this, and we're just trying to understand how this pan out. And in the future, we're gonna have more specific data on this. But yeah.
The data you have shown on the biomarkers of inflammation in response to methionine supplementation, may I know at what time point these samples were collected related to feeding?
Yeah. So yeah, all the samples we normally do before feeding. So yeah, we don't have that potential effect by having a different sampling 3-4 hours after feeding. So we're always sampling before feeding, so we account for this. Yeah.
Thank you very much. Sorry.
Okay, thanks.